So we start with our next topic, cell-free fetal DNA. Okay, this is a talk which is published in 2017. Again, on this particular talk, there can be single best answer question. So what is the difference between non-invasive prenatal test and non-invasive prenatal diagnosis? These two terminologies which you have to understand. Okay, because sometimes in part 2 they will ask you SBA and whether to select answer as NIPT or NIPD. Out of 10 or 12 options, you know that it is a prenatal test, non-invasive prenatal, but whether it is called as NIPT or whether to select NIPD. Okay, so non-invasive prenatal testing, NIPT, uses cell-free fetal DNA to test for aneuploidy, as opposed to non-invasive prenatal diagnosis, which uses cell-free fetal DNA to diagnose fetal sex, RH status, and monogenic disorders. Okay, so in both cases, cell-free fetal DNA is used, but NIPT is used for aneuploidy, while NIPD, that is diagnosing the sex, RH status, and monogenic disorders, that comes under NIPD. Basically, NIPT is a screening test and it needs a confirmatory invasive testing in cases of high risk or positive result. So NIPT is a screening test which will be followed by the confirmatory invasive testing, while NIPD is a direct diagnosis. Okay, there is a diagnosis of fetal sex, there is a diagnosis of RH status or the monogenic disorder. That is what is the difference between NIPD and NIPT. Okay, so NIPT demonstrates high sensitivities and specificities according to recent meta-analysis, although it is less accurate for trisomy 18, 13, monosomy X, and sex chromosomal aneuploidies than the trisomy 21. So highest sensitivity is seen for trisomy 21 and less for other trisomies like 18, 13, monosomy X, and sex chromosomal aneuploidies. Okay, we will see further in the table in the next slide. So what is the basis of NIPT technique? So cell-free fetal DNA comprises small fragments of fetal DNA thought to originate from the trophoblast. These fragments circulate in the maternal plasma and form approximately 10% of DNA fragments in the maternal plasma. So almost 10% of the DNA in the maternal plasma is from fetal origin. It is present in reliably measurable levels from 10 weeks of gestation. This is a SBA. At what weeks of gestation it starts in the maternal presence is seen in the maternal circulation around 10 weeks of gestation. It is cleared quickly from the maternal circulation hours after delivery, making it specific to that pregnancy. Now, what is the test accuracy? NIPT for trisomy 21 and trisomy 18 demonstrated high sensitivity and high specificity. Results of trisomy 13 revealed a lower sensitivity. Exact reasons for this remain unclear. It is thought to be associated with low guanine cytosine content known to exist in chromosome 13. Monosomy X demonstrated reduced sensitivity compared to trisomy 21 and 18. So if you see the sensitivities of this particular test in this particular table, you will see that it is highly sensitive for trisomy 18. Okay, so see under the heading of sensitivity, that is 0.994, that is 99% sensitive for trisomy 21, then 97.7% for 18 trisomy 18, then 90% 0.906, that is 90% sensitivity for trisomy 13, and monosomy X 0.92. Okay, so highest sensitivity is seen in trisomy 21, followed by 18, followed by monosomy X, and followed by 13. Okay, so in multiple pregnancy, trisomy 21 was detected at a rate of 93.7%. Just now we have seen the highest sensitivities for trisomy 21 for a singleton fetus, that is 99.4. But for multiple pregnancy, it is 93.7%. And false positive rate will also 0.23% in twin pregnancies. Okay, demonstrating lower sensitivity than testing in the singleton pregnancies. Okay. Now, there are problems in the twins. Okay, when you perform these particular NIPT, there are problems in the twins. So what are these problems? Suppose you have a dizygotic twin. Aneuploidy discordance is an issue and nearly a twofold difference in the cell-free fetal DNA fraction. Affected fetus may have a cell-free fetal DNA fraction below the threshold of 4% required for testing, while unaffected twin may contribute to high cell-free fetal DNA fraction. Therefore, the total cell-free fetal DNA fraction may appear sufficient and produce false negative result. See what happens, the minimum required fraction for test, uh, uh, test to carry out is a 4%. And when you have a dizygotic twins, in that particular case there may be one fetus which is affected, the other fetus is normal. But if the affected fetus is not contributing to the cell-free fetal DNA much, if the critical level is 4% required to detect, but if the contribution is less than 4%, then it will not be detected, while the normal fetus will contribute it more and it will be reported as a normal. Okay, you understood this?
Yes, sir.
Okay,
Yes,
so
sir.
so this can be a problem in dizygotic twins. Testing in monozygotic twins, theoretically it should be easy. Okay, because the identical DNA molecules will be produced, but one should be very sure about the chronicity. It is a monozygotic twin. Okay, so monozygotic twins, they will have an identical genetic material, while in dizygotic twins there will be discordance in the genetic material, but the quantity of the affected gene, if it is adequate, then only it will be picked up. Okay. Another problem is that of a single twin demise. As the effect that CL-free fetal DNA from the demise twin has on the NIPT's result is unknown. Sometimes what happens, there is a single twin demise. But if there is a contribution from that particular fetus where there is a demise, in for the cell-free fetal DNA, so that what exactly the result is still unknown. Because of this, professional bodies do not currently recommend NIPT for aneuploidy in twin pregnancies, including RCOG or ACOG. So NIPT is not recommended for aneuploidy diagnosis in multiple pregnancies by the RCOG and ACOG. However, in private, it is available even in UK, but it is not recommended by these professional bodies. Okay. Now coming to the benefits of NIPT. So it is a non-invasive test. So it does not pose the risk of CVS or amnio such as pain, small risk of infection, procedure-related miscarriage, which are associated with CVS and amnio. Okay. Then cell-free fetal DNA is cleared quickly from maternal circulation. It is specific to that pregnancy. The test has a quick processing time, reported in 3 to 5 working days, equivalent to QFPCR, that is quantitative fluorescence polymerase chain reaction testing for invasive samples. Okay, so quick processing time. So these are the advantages of or benefits of NIPT. That first thing, it avoids the invasive test. Second, it is quickly cleared, so it is specific for that particular pregnancy, and rapid reporting is available. Now, what are the disadvantages of NIPT? So test is not 100% accurate. Okay, there are false negative and false positive results. Occasion test will not produce. Sometimes the test will not give a result. That is inconclusive test is the result. So inconclusive result is seen in 1.9 to 6.4% of samples. So approximately 2 to 6% there will be inconclusive result. Who have inconclusive results are more likely to have chromosomal aberrations. Okay, so this is also important. If you get an inconclusive result, but among these inconclusive results, there are high chances that most of the time there will be chromosomal aberration. Okay, so what are the common reasons for false and inconclusive result? One is a low fetal fraction in the blood sample, which is measured by specific markers of fetal DNA or algorithms which are applied for sequencing the data. Okay, so fetal fraction, if it is low, it will give negative result. Second is a vanishing twin that has disappeared prior to the woman's dating ultrasound scan, which, if non-identical, may cause false positive result. Okay, so if it is a non-identical twin and there is a vanishing twin, it has disappeared, but the cell-free fetal DNA is present in that particular case, there can be a false result. Okay. Then confined placental mosaicism. Sometimes what happens, the fetus and the placenta, they have a different lineages. The fetal DNA fragments originate from the placenta. The NIPT is unable to distinguish between the two. Okay, so there are two lineages, two separate things. Sometimes there is a placental mosaicism, and that is not detected by this. Okay, so NIPT can detect maternal cancers and maternal copy number variants, which result in false positive and have ethical implications. Sometimes there can be genetic material which is from the cancer cells, okay, not from the fetus, but it is picked up as an abnormal thing. Okay, so that can cause a dilemma also, ethical dilemma also. And NIPT has an effect on fetal medicine specialists because there is a reduction in the invasive tests which are carried. So almost 53% reduction in the amniocentesis and 77% reduction in the CVS, which has happened because of the availability of this test. So that finishes the topic of CVS and amnio. Thank you.